So what is remote sensing? One definition that I found according to this National Ocean and Atmosphere Administration in the US is that remote sensing is the science of obtaining information about objects or areas from a distance, typically from aircraft or satellites. So if we consider remote sensing in its broad meaning, this is something that we perform every day. I mean, sight is basically something about remote sensing, what we smell, what we hear, and there's lots of animals that have developed different senses that are related to remote sensing, like bats and dolphins using echolocations, or dogs smelling at distance and cats seeing at night. So one other way to define remote sensing is to say that it's about quantifying the interaction of light and matter. And in the case of what we are working on, matter is of course vegetation and crops, but it can be a number of things from stars, galaxies, or gas, oceans, buildings, group of animals, virtually everything. So then, what is light? Well, as you probably know, light is basically some energy that is emitted by excited electrons in the sun or in a light bulb. And to simplify a bit, we can see light, or we can consider light as a wave. And then light colors can be thought as wave with specific frequencies. For instance, you can see here that these waves, these purple waves, have higher frequencies than the red ones. And all these waves actually define what we can see with our eyes, the visible light that we can see like with rainbows. But the thing is that there's much more information out there than what we can see with our eyes. So here, what you can see is the actual complete light spectrum. And here you have the very, very high frequency wavelengths, like cosmic rays and gamma rays. That's a matter of 10 exponent minus 12 meters. So that's like really small. And on the other end, you have the long wave bands, like radio or broadcast bands, which are long of several kilometers. And here is actually what we can see with our eyes. So that's really a very tiny part of the actual light that we are capable of sensing with our eyes. And that comes from some evolutionary reasons. We ended up being sensitive to these specific colors, but some animals had different evolutionary paths. So for instance, bees and more generally pollinators happen to see in part of the ultraviolet range, and that's very convenient because there's much information that they can get from flowers. In this case, this dandelion, we see it as yellow, but a bee will see where the pistil, where the pollen, and where the nectar is much more accurately. So that's an evident evolutionary asset. And on the other end of what we can see, past the red part of our spectrum, there's the infrared, and some snakes can actually sense in the infrared. And that's also a bigger set when hunting during the night to spot prey based on the temperature that they have. So then, the point is that there's lots of information out there that we can use to get a better understanding of what's going on with vegetation. That's one reason for using remote sensing, but there's also a couple of other important reasons. First one is that it's based on objective measurements. I mean, remote sensing is based on physical laws, and these laws are well known and established, and then we can repeat our measurement in different time, different spaces. We can compare things. Second reason, as I was saying, we can compare through time and space. So here in this case, you see a set of satellite images that have been taken over the Netherlands. And you see sometimes they are like red plots that appear. That's tulips blooming in the Netherlands. So we can track the phenology of crops using remote sensing, for instance. And last but not least, this is a non-destructive measurement, which means that you do not need to, if you want to get information about your field or samples from your field, you do not need to walk into the field, harvest the sample, take it back to the lab, analyze it, and process these results. You just get an image or you just get some data and you can get the results very rapidly and without destroying any part of the plot or field. So then, how do we use remote sensing? The basic principle is more or less always the same. We've got a source of light here. This can be the sun, this can be a light bulb, and this source of light will emit a ray that will go up till a target, which in our case is a plant, and part of the light will be absorbed by the plant. Part of the light will go through the plant and hit the ground and then bounces back. And part of the light will actually get reflected by the plant. And this is this light that we're going to measure with different sensors that can be mounted on drones or satellites. So in the end, in our case, remote sensing of vegetation is about taking advantages of the information contained in the light spectrum, especially in the ranges that we are not able to see with our bare eyes. We usually look at what is happening from 400 nanometers here to 2500 nanometers. These are like the part of the spectrum that contain all the information that is relevant for us about vegetation. And so this is a typical spectrum of leaf or canopy. You have here the amount of light that's reflected. And now we can split this spectrum into 3 different parts. So the first one is what we call the visible range. As I've explained, this is what we can see with our bare eyes. And it goes from 400 to 700 nanometers. And this gives us information about the pigment content in the leaf or in the canopy. And for instance, you see here this little peak of reflectance. It means that more light is reflected here. And this is actually the green part of the spectrum. So this is the greenness of the plant that we are seeing here. And here is the blue, here is the red. So yeah, most of the light is absorbed in these wave bands. The second part of the light spectrum that is of interest for us is what we call the near-infrared range. Because, well, the visible range is related to colors of the plant, so this is related to pigment content. But the near-infrared range is sensitive mostly to the biomass of the plant. So the more leaf and the more the canopy develops, the higher the reflectance in the near-infrared range will increase, and this is information that's very interesting for us. And finally, there's what we call the shortwave infrared range here, and it's mostly related to the water content of the plant with these depressions here and here, and the protein contents. So in the previous slide, I was talking about drones and satellites. These are like the most widely used tools for remote sensing. Also, there's also some ground-based sensors, but I will come back to those ones a bit later. So drones and satellites, they are mostly doing the same thing. They're giving us pictures with spectral information that we can use to track changes of fields over time and estimate different traits that are of interest for us, like biomass or nitrogen content of leaves. The big difference between drones and satellites is the spatial extent and the spatial resolution that we can get out of these systems. Drones, for instance, are very handy if you want to work at the field scale or at field experiment scale, because it will be fairly easy to get a very good image of something like 4 or 5 hectares within a flight of 30 minutes. And you will get a very good spatial resolution. The image that you will have pixels that will be within the centimetric scale, can be a bit less, can be a bit more, depending on the sensor. But then you have a very small spatial extent, which means that you cannot apply this solution to large regions. On the other end, we have the satellite remote sensing, and this is definitely something that we want to use when working at large scales, like northern Sweden, for instance, looking at one glance at all the different fields growing in northern Sweden. And the drawback then is that it only has a spatial resolution that's within the metric scale. Most of the satellites that we are using have spatial resolution ranging between 10 and 20 meters. So to illustrate that a bit more, this is an example of a drone image that was acquired in LENAS last year. And now if we look at the equivalent of this image from a satellite sensor, this is what it would look like. So you see we lose lots of spatial information. We actually don't see the differences between the different plots. But the thing is that this is only a part of a satellite image. So yeah, when dealing with remote sensing, it's very important to frame the sensor that we want to use depending on what we want to assess. One of the things that's quite important with remote sensing that you might have heard about is the different type of sensors. So the first type of sensor is what we call the RGB sensor, and that's what you will have on a regular camera, like the one on your phone, or and this is equivalent to what we can see with our eyes. Basically, it's using a sensor that's sensitive to the blue, to the green, and to the red, and then by combining these different informations, we can reconstruct all the colors that we're able to see. Then there's also multispectral sensors. So the big difference is that it's not only into the visible range, this is also including bands into the near infrared range. And this can have bands like from 3 bands to some sensors up to 20 bands, which will be measuring the reflected light on all the range of this light spectrum. And then there's what we call the hyperspectral sensors. These are not really, how should I put it? We cannot really use them out of research. This is still a tool that's for researchers, but I think that within the next couple of years, this will become more and more used for farmers as well, because it contains lots of very interesting spectral information. It's a bit tricky to use, but it definitely yields very good results for looking at different characteristics of the plant. So now that you know a bit more about remote sensing, I would like to tell you a bit more of the research that we are conducting at my department. And I would like to start with the satellite remote sensing. So there's lots of different satellites out there that can be used for Earth observation and looking at vegetation, tracking changes of crops. But there's one that's like really interesting in my opinion. It's the Sentinel-2. So this Sentinel-2 has been launched a few years ago by the European Space Agency. And it's actually a constellation of 2 satellites that are orbiting the Earth at opposite directions. It's really interesting to use for us for several reasons. First one is that it has a fairly decent spatial resolution from 10 to 60 meters, depending on which bands we're looking at. It has 13 spectral bands ranging from the visible and also in the near infrared and short-wave infrared range. So we can get lots of spectral information that's relevant for crops monitoring. It's also processed by the European Space Agency, which is really good for any lazy researcher, because then we don't need to worry about applying some corrections about the geometry or the radiometry of the image. We can just use them as they are. And last but not least, those satellites, they can provide images up in the north of Sweden every second day. So we can have a very good tracking of changes of vegetation by looking at what's happening every second day. And also they're open access, which means that anybody can access this data, download them, and use them. We do not need to pay any fee for them. And one of the research projects that we are working on at the moment, that's an ongoing research project, which is called the Vallsat project. And the idea is to use Sentinel-2 images to perform near real-time estimation of leys traits at the field level. And the traits that we're looking at are the dry matter yield, the nitrogen uptake, the crude protein concentration, and the digestibility. So as I said, this is an ongoing project. We're still doing measurements this specific year and hopefully also during the next years. And the idea will be that with the models that we'll develop in this project, we'll be able to provide farmers with a system to help them to decide on when to harvest based on the biomass accumulation and the digestibility of the crop, recommendations with fertilization based on the nitrogen uptake, and so on. So we want to build a decision support tool to help farmers to manage their grasslands in the best of possible ways. So yeah, we're taking measurements on 4 different sites in the north of Sweden. One is in Ås. We've got another site at Lennäs, and we've got several fields at Röbäcksdalen, close to Umeå. And we also have, or we had, one field in Öjebyn for tracking, performing measurements. So we're still working on different approaches here, but there is one in particular that I would like to show you. The idea is basically to take advantage of the high frequency of those Sentinel-2 image acquisition that I was mentioning. So what you see here is the changes that are happening in the fields of Röbäcksdalen between 2019 and 2020. And we can clearly see the differences of what's happening in the field when they've been harvested, when they started to grow again, etc. And this is this information that we're interested in. So the idea is to compute a vegetation index based on these satellite images, and using those we can track more accurately the changes and quantify the biomass accumulation, for instance. So vegetation indices, this is also something that's quite important in remote sensing of vegetation. Basically, a vegetation index is about combining 2 or more spectral bands from a sensor, and so that we can extract relevant information for us. And the most famous vegetation index maybe is this NDVI, which stands for Normalized Difference Vegetation Index. And basically this is computed using the red information and the near infrared information. The red because, well, when the plant has lots of chlorophyll and that everything is going fine for the plant, there's very little reflectance into the red. And on the other end, when the plant is healthy, then there's lots of light that's reflected from the infrared range. So this is where we can quantify the most easily the differences between stressed and non-stressed plants. So for instance, if we compare the stressed leaf here and the healthy leaf, we can clearly see that there's those differences that I was mentioning about reflectance in the red. Here there's less light that's reflected when the plant is healthy. And in the near infrared, there's more light that's reflected when the plant is healthy. And that induces that when the plant is healthy, the NDVI is going toward 1. And when the plant is stressed or dying, it's going towards 0. So there's two big advantages with these vegetation indices. The first one is that it helps to discriminate vegetation very easily, actually at a glance. Because for instance here, you see this image, this is over the field in Röbäcksdalen, so this is Umeå. And here is the equivalent of the NDVI of this image. And all that's white, all that's appearing white is not vegetation. And what's appearing very dark black is actually vegetation that's doing fine. And you can see, for instance, that there are some differences that appear to be more obvious when looking at the NDVI image. For instance, here, this field, if you look at the original RGB image, the difference is much more difficult to spot. So if we apply these NDVI computations to these satellite images that we've been acquiring over Röbäcksdalen, for instance, here you can see the changes through time. And if we focus on one particular field, say for instance, this one, we can track the changes through time and build what we call NDVI profiles. And for instance, here, if we look at this field, we can clearly see how the NDVI increases through time, and this corresponds to an accumulation of biomass. And then here we have the first cut with the sharp decrease of the NDVI value, and then the second growth starts over. We reach a plateau, and then there's the second cut, and then biomass starts to accumulate again, and then there's the last cut, and then it's winter and plant is just dying. So how do we convert this information to an estimation of the biomass production? So the hypothesis is that the accumulation of NDVI is a function of the accumulation of biomass. So if we sum the NDVI between two cuts, for instance, we should be able to retrieve the biomass with that. And so what we do is that between these two cuts, we'll compute the equivalent of the area of this surface. And we will do that for all the fields and all the cuts of each field. And finally, we will try to link that to biomass, these summed NDVI values. And this is the type of results that we got so far. Here you have what we call the predicted dry matter yield. That's the one that's derived from the satellite information. And here we have, well, observed, that's actually the measured dry matter yield. So this is expressed in kilograms per hectare from 0 to 6,000. And we can see that we've got a fairly good linear regression, which is very good for us because this is what we're looking for. And so we are trying to get more data to test this this approach and see if this is definitely something that we can use for as a tool for helping farmers, or if we'll find some limits when adding new data. So we are still trying to test this solution. Unfortunately, I don't have any results to show you about the estimation of the quality traits of the plant, like the digestibility or the crude protein concentration. But there's other studies that show that there's a fairly reasonable good potential for Sentinel-2 to retrieve quality traits of grasslands. So you see here, there's not lots of points, but I mean, still, that's a result. also it's been acquired for different climatic contexts, because this is for the Mediterranean region. But you can see that for the crude protein concentration and the fiber content, we've got regressions that are fairly okay, and we're hoping to do at least as good as what those people did, and hopefully maybe better. We've got lots of ideas to test. I would like to wrap up this satellite remote sensing demo with an example of some kind of practical example for fertilization rates. And maybe some of you have heard about this CropSat solution. This is a tool that's been developed by DataVext with SLU. And let's see if I can. So if you do not see my screen, just let me know. So maybe you're familiar about this website, but basically it's using satellite images to perform recommendations on amounts of nitrogens to use. So if we go, for instance, to these fields in Röbäcksdalen, and we can look for blocks, say this one, and then the platform will look for satellite images that have been acquired recently or within a certain period of time. And here we have this picture which has been taken on the 24th of August. And then we can look at recommended strategies. The platform is computing indices, vegetation indices that can be related to the need of nitrogen for the plant. And here you get this map. You see where the field actually needs some more nitrogen and where it does not. For instance, those yellow pixels, this is where there's a deficit in nitrogen, or maybe not a deficit, but not as much as nitrogen as there should be. And the green area is where, well, the crop is doing fine and maybe it's not necessary to put more nitrogen. And then here comes the last part. It's related to your knowledge of your field. You can adjust the amount of nitrogen that you want to put based on these values of this vegetation index. So say you could put 90 here, 85, 70, and then for these ones, drop to 20 and 15, something like that. And then you can get a file that you could upload to your nitrogen fertilization system, and then it will adapt the fertilization rate depending on the recommendations built here in this tool. So that's definitely what we're trying to aim for with the Vallsat project. This has been developed for cereals, but we want to make it working as well for leys and grasslands. So now that we're done with satellite examples, I'm going to talk a bit about the research that we are conducting with drone remote sensing. So just as a reminder, the only real difference between satellites and drones is that we are not getting the same spatial resolution. So when working with one single field or working with field experiment, it's very relevant to use drones. And here you can see this is the type of image that we'll get from the drone images. This is actually Bali plots at Röbäcksdalen in Umeå, and you can see that there's different cultivars just based on the color. And there's actually one thing that we can do with drone that's not like super easy to do with satellites. It's building 3D models. So yeah, that's the kind of thing that we can get with a drone. So we had a drone flying over this field experiment, taking lots of images. I think this was like 300 images. And then we throw these images into a dedicated software that will stitch all the images together and apply some photogrammetry principles that will allow to build for these 3D models. And that's really interesting for us for several reasons, because by doing this, we are getting information about the colors of the crops, which is indeed in itself quite interesting, because we can relate that to chlorophyll and through chlorophyll to nitrogen content. But we can also get information about the height of the crops. And height is a direct proxy for the biomass production of a crop. And the biomass itself is a direct proxy of the grain production of a crop. So we can actually estimate the grain yield of plots using this approach. So that's what we are trying to do with one of our projects. This is not about barley, but this is about red clover. We've got trials of red clover that we use for breeding. And we want to look at their biomass production, if there's any difference between the cultivars. So here is a 2D image. That's the equivalent of these 3D models that I was showing you. So the white objects are like tall ones. Here, for instance, you have the car, and here you have trees, and here are the plots of red clover. And on the other end, the objects with dark pixels are small objects. So that means that we can compute the height of the crops. So if we look, for instance, at this specific plot, we can extract all the pixels that correspond to this plot and compute the average of the height, and then relate the height to the biomass. And that's what we did here. We've got these results for measurements that were made in Linus. And you can see that there's a fairly good, not completely linear relationship between the predicted fresh yield, that's the one derived from those 3D models, and the one that was measured in the lab. So that's also an ongoing project. We've got lots of data that we still need to process, but we've got some encouraging results for that. And once again, this can be applied, well, this is applied here for red clover, but this can be applied to cereals as well. So I'm not spending lots of time on drone applications. There's lots of them, but I mean, it overlaps quite a lot then with remote sensing. What I did not mention yet is how ground-based remote sensing can be used. And I would like to focus on one specific, very specific aspect of what we can do with ground-based remote sensing, and that is the estimation of the clover content in grasslands. And I'm going to show you two different approaches that we've been testing and that we are currently working on. So the first one is clover content estimation with a Yara-N sensor. So I would suppose that most of you are familiar with this type of sensor. It's been basically developed by Yara for recommendation of fertilization rates for cereals. But then we thought, well, why only cereals? Maybe it will be worth trying it with grasslands, because, well, the light information is the same, what we need. So the Yara-N sensor is usually mounted on tractors. That's a big, heavy one that we cannot really easily use on the field. So we had a different one. I mean, the sensor itself is the same, but it's a handheld version, smaller, much lighter version of the Yara-N sensor. And here you can see there's a sensor that's on top of this instrument that will measure the light that comes from the sun. And there's another sensor here that's measuring the light that's reflected by vegetation. So this type of tool, this does not give any image. It just looks at the amount of light that's reflected from an area and sees it as a single pixel but it has very rich spectral information. For instance, this is typically what we get from this Yara-N sensor. So here you have the wavelength from 400 nanometers to 1000 nanometers. So the Yara-N sensor looks at light into the visible range and partly into the near infrared range. And what we did was to use this instrument on different field trials and plots with different species of grasses, different species of clover, and see how it can be used for retrieving the clover concentration. So we basically took some readings with the sensor and then cut the samples, go back to the lab and separate what was grass from what was clover from what was wheat. And then we used statistical models. I'm not going into the details of these models, but there were two of them. The first one is this PLSR, which stands for partial least square regression. And the second one is SVR, which stands for support vector regression. This one is actually part of the models that are called machine learning. And so here on these graphs, you can see on the x-axis, this is the prediction of the botanical composition. So once again, what's estimated from the spectral information that we got from the sensor. And on the y-axis, this is the observed botanical composition. And well, you can see that both models are yielding fairly accurate results, but there's big differences between the models. In the case of the PLS, there's lots of scattering of the data around this regression line, which is, I mean, it's still reasonable in terms of accuracy, but we would like to squeeze and reduce the scattering of this data as much as possible. And then when we look at the results for the support vector regression, you can see that there's much less scattering around the regression line. So this is what we're aiming for. And we're actually, well, we've been conducting this experiment starting in 2017, if I'm correct, up till 2019. The results that you see here are only for 2017. So we'll test these approaches for the rest of the years. And if it indeed works, we'll try to find a way to make that available for farmers that already own a Yara-N sensor, because then, depending on the amount of clover that's on the ley, you might want to adjust the rate of fertilization. The second approach that we used is based on deep learning. So I guess that you all heard about deep learning. This is a statistical approach which allows for image analysis and the recognition of patterns like people, cars, cats, signs, whatever. And here we've applied that to the recognition of clover into RGB images. So the idea was basically to take pictures with a smartphone, regular RGB images, and then throw that into the model and see how it can recognize clover. So these are the original images that you can see here with low clover content, medium clover content, and high clover content. Then there has been a huge work of doing a manual labeling of what's clover. So you can see here, this is like grass is in purple and clover is in blue. And you can see here the clover appearing very clearly. And then we just threw that to these deep learning algorithms, and they learn themselves a way to recognize the clover based on the color, based on the texture, based on the shapes of the clover. And you can see that, so these three different deep learning algorithms, the deep lab one, the SegNet one, and the FCN one, they're yielding different results. But overall, the one that's doing better is this deep lab v3+, and it has a fairly reasonable accuracy. If I remember correctly, it's like 95% of correct recognition of Clover. So that means that we're able to propose a solution based on images taken from your phone to tell very rapidly what's the amount of Clover into grasslands. So to wrap up this presentation, I would like to just say a few words about our research facilities, the one that we have at SLU, because we started to build a spectral lab at Röbäcksdalen. We've got lots of different equipment. We've got different drones with RGB and multispectral sensors. We've got this hyperspectral camera. This is something that I did not mention in the talk, but it's not very relevant for field monitoring, but it has very nice applications on quality of food or of discriminating different types of vegetations. And we also have field spectrometers. So I do not show the Yara-N sensor here because you already know it, but we've got this field spec, which is actually even more accurate and exhaustive than the Yara-N sensor. We want to use it as much as possible for developing solutions for farmers. And we also have this Link Square, which is a compact spectrometer that's very easy to use and quite cheap. So we are looking at how it could be used as well for farmers to get information about their crop in a very quick and accurate way. So I guess my message here is, if you have any questions, if you have any suggestions of what type of research will be interesting for farmers or advisors to help them doing their job, please come to us and let us know. We are very happy to have this kind of feedback to decide on which research to work on. And well, to finish, I just would like to briefly acknowledge different people and organizations here, because all the work that I presented was funded by these different organisms. I'm sorry for my accent, but there's the regional jordbruksforskning in Norra Sverige, Sveriges landbruksforskning, and Lantmännen funding research, and also the Campay Foundation. And also, I would like to thank the people at the field station and sites, which is a network for monitoring environment in Sweden. Well, that's it. Thanks a lot for watching. I hope you enjoyed.